Hey everyone, thanks for joining. I'm your host, Sean Opie, and today we'll be talking about minimal residual disease testing in lymphoma. This is intended to be a kind of a medical science style presentation. And so what we'll be talking about is a clinical trial. The results were published in the Journal of Clinical Oncology 2021. This comes out of the Frank and Miklos group. They're Stanford. And I always like to provide the abstract just so that if somebody wants to go and get the paper for themselves, they have all of the information that they need. So this is what we're going to be talking about today. Since we started the title with minimal residual disease, I thought we should talk about this briefly. It's defined as the presence of a small number of cancer cells that remain in a patient's body during or after treatment, even when the patient appears to be in remission. And so what this means is that there just are not enough cells or enough cancer cells to be able to be seen by traditional methodologies such as PET-CT for metabolic imaging, IHC, immunostaining, and even flow cytometry. And the problem with these cells is that there's just enough of them to cause, or at least to potentially cause, relapse. And as a patient, that's a very bad thing to hear. So on the right-hand side, we have a figure that I pulled from a Van Dongen 2015. I think it's a really good description of MRD. You can see on the y-axis, we have the relative frequency of lymphoma cells. This is just a surrogate for genomes or DNA molecules, and then arbitrary time points on the bottom. And immediately following treatment, we should see a precipitous drop in the frequency of lymphoma cells, but these can really segregate into three different categories. Early relapse, where treatment was basically ineffective. Somewhere below your typical cytomorphology detection limit, relapse can occur. It just takes a little longer. Again, the cells have to multiply so that they become visible and a relapse diagnosis can be made. And then finally, there's this below the 10 to the fifth or 10 to the minus fifth line. This is where we get into the area of minimal residual disease. And there are a variety of companies that are trying to, or that are making assays capable of detecting down to 1 in 10 to the sixth genomes per milliliter of blood, which is quite remarkable. And so that is what we are going to talk about today, MRD testing, which brings us to the value of it. And I think this is just a corollary from the other slide, but it's going to help early detection of disease progression, which means that it can help predict relapse. When done in the context of clinical trials, it can help with personalized treatment approaches and certainly support both industry-sponsored and investigator-initiated clinical trials for both on-label and new investigational drugs. So because this presentation was prepared at the request of Adaptive Biotechnologies, they're a Seattle-based diagnostic company that really specializes in hematological malignancies. I thought it would be worth taking a little bit of time to talk about their clonoSEQ assay. It's a next-generation sequencing based, so NGS-based assay, and using sequencing, it identifies and quantifies. I think that quantifying part is very important. Rearrangements in the immunoglobulin genes, and also a couple of translocations that are critical for lymphoma, BCL1 and BCL2. I like to remind people that this measures cell-free DNA. It doesn't measure actual cells. Again, this is a nucleic acid-based test. It's currently cleared by the FDA as an IVD for use in B-cell acute lymphoblastic leukemia, multiple myeloma, and chronic lymphocytic leukemia. And so this particular trial that was run is part of their support for trying to get IVD status in lymphoma. Now, it's not available as a CLIA-validated test, but these can still be run as an LDT for academic or even physicians that are trying to get additional information with respect to treatment. So Just a reminder that not everything has to have IVD status to be helpful. The cell-free DNA collection process is pretty simple. There's some peripheral blood collected just in a regular tube. It's actually a cell-free DNA tube. They separate plasma. They take those aliquots and store them, and then this plasma is where the cell-free DNA is extracted from, and then it just goes on to typical next-generation sequencing. This is a fairly standard set of laboratory instrumentations, and importantly, it's relatively non-invasive. It's just a tube of blood from the patient's perspective. Okay, getting back to the patient, or getting back to the paper now, I always like to ask, what is the question of the study? And here, the question is, does minimal residual disease testing have prognostic value in patients with relapsed or refractory large B-cell lymphoma treated with axicabtagene or Yescarta? So that's just a CAR T autologous cell therapy. Now remember, this test, the clonoSEQ assay, is not IVD. It doesn't have IVD approval or IVD status for lymphoma. So that's part of why this study is being generated. And to reach that, they're setting up two endpoints. The first, and I'm just going to read these verbatim from the paper because these are very, very important. First is the ability of ctDNA-based assessments at the time of starting lymphodepletion to predict progression-free survival of patients with large B-cell lymphoma undergoing CAR T therapy with at least a 6-month follow-up. So there's got to be 6 months of data before we can make any prognostic assessments. And then the second one is the association of ctDNA-based MRD assessments on day 28. That's usually when PET CTs are done to see to get some staging data. On day 28 to predict progression-free survival status of patients with LBCL undergoing CAR T therapy. So 2 questions. Okay, here we go. The study puts forth a consort diagram. It's a relatively simple single-arm study. They enrolled 72 patients. Here are the most important characteristics of these patients. Had to be older than 18, have a diffuse LBCL, couple other kinds included. They needed to be enrolled before lymphodepletion for CAR T cell therapy. They had to have a tumor block available so that they could do some clonotyping, which is just part of this assay. And then they needed to have measurable PET avid disease. So they have to have hot spots on PET CT. Of those 72 patients, you can see that 8 of them were excluded. 3 of them had no dominant clone. I'll talk about that in just a second. 3 died before day 28 related to CAR therapy. 1 patient progressed before day 28. Remember, there was a time requirement in here. And then 1 patient was lost to follow-up before day 28, leaving 64 patients for final analysis. The demographics are presented in the first table. There's a couple definitions we need to look at: a durable responder and a progressor. So a progressor was defined by the study as a participant who experienced a progression-free survival event. That usually means some form of disease progression or relapse, right? A progressor. And then a durable responder. This is a patient who did not experience a PFS event with at least a 6-month follow-up. Couple interesting things to look at here. First, the statistics that are provided here with p-values are comparing durable responders and progressors. This is after the study data is available. So we couldn't do this kind of analysis at the onset of the study because we didn't know the progressor or responder status of these patients. So it's just first thing. The second is there are some differences here. First, it looks as though our progressors are generally younger. And so, likewise, the responders are generally older. There appears to be a very important statistical difference in an elevated LDH. So lactate dehydrogenase is actually an independent predictor of progression, but that's been shown in a variety of different studies, not just this one. And then finally, they have some statistics about the number of people with double hits. So those are patients that have 2 or more mutations that put them at risk. And I'll talk about these a little later in the presentation, but in both, for the total enrolled patients, this is higher than would be expected. You know, traditionally in the general population with this disease, 5 to maybe 10, maybe 15% have double hits. So, you know, it's just important to know that there were more patients than would have been expected with the double hit. But otherwise, these patients are relatively, they're very comparable. Okay, so the first thing they did was use the test to identify clonotypes or specific rearrangements in these immunoglobulin genes. And you know, this is not a presentation about the assay, this is about the medical science. So we're not going to geek out about the assay itself. But again, it's looking for VDJ recombination events. And what we can see here is that the number of trackable sequences goes from 0. Remember, those are 3 patients that were excluded because they had no specific B cell clonotype identified, all the way as high as, or as many as up to 6 different clonotypes were identified. You know, we can translate that and say that 96% of these assays provided some kind of information to the physician, and only 4% the test really couldn't be used. So it's very applicable, and it's providing good data, providing usable data. So here we get to our first figure. We're going to take a look at some representative patients. Figure 1A, we start with a patient that is in a complete remission. Now that's determined by metabolic imaging and also considered MRD negative. And so over here in 1A, we can see that this patient started with 180 plus lymphoma genomes per mil of plasma. This is where lymphodepletion starts. At day 7, you can see they are down to being MRD minus. And throughout the course of all the follow-up, which is about a year, molecular testing never identified any residual disease. Here's the PET-CT data. You can see that pre-treatment, there's hot spots all throughout the lungs. At day 28, they are gone, so they are in a complete remission. And the same thing continues through day 365. So again, this is just imaging data that really goes along very nicely with the molecular testing data. This is in contrast to figure 1B, which has a patient in a PR post-treatment and also never gets to MRD negativity. So you can see this patient has 17, almost 18,000 lymphoma genomes per mil before they start lymphodepletion. They are blasted with chemotherapy, and you can see they still have somewhere between 800 and 1,000 lymphoma genomes per mil of plasma. And at no point along their timeline, which goes out to 90 days, are they ever MRD negative. So again, imaging data, we can see a fair bit of disease. At day 28, there are still hot spots here. So this person is, while they have, they have responded, but they are still in partial remission. And unfortunately, by the time day 90 rolls around, you can see that disease has really exploded. And this is full-on progression, full-on progressive disease. So again, looking at molecular data and how that compares to imaging data. The next figure 1C talks about the time course of the median concentration of ctDNA, comparing durable responders to progressors. Median because we're talking about log style numbers here, so it just makes sense to do a median rather than a mean. Otherwise, they'd be skewed in one. They'd clearly be skewed. This chart's a little busy, but Again, on the y-axis, we have lymphoma genomes per mil of plasma. Note that it's in a log scale. I also want to put or note this 0. So this is minimal residual disease negative, and there appear to be 1 or possibly 2 dots along the bottom. But it's important to draw your attention to these no detectable ctDNAs. So there are actually 9 blue dots here and 3 blue dots here. And of course, that goes all the way along. What we can do is do some math and calculate an area under the curve, which basically compares any cohort of blue dots in one vertical line to its corresponding cohort in the red line, red dots on the other side. And excuse me for not pointing out that blue are your durable responders and red are your progressors. So even with just an eyeballing, you can see that the durable responders have a lower number of genomes per mil than do the progressors pre-lymphodepletion. But the area under the curve really mathematically calculates these. And in a clinical setting, generally speaking, anytime you have an AUC at 80 or above, it's considered clinically significant. And that lines up with all of these p-values here. So the simplistic analysis of this is that progressors generally have a statistically higher count of lymphoma genomes per mil of plasma than do durable responders. And again, just a reminder to everybody that these counts here are representative of the number of dots that are supposed to be here. Okay, let's move on to some Kaplan-Meier curves then. And we're going to look at all patients, these are all patients, progression-free survival and overall survival by their initial pre-lymphodepletion ctDNA concentration. So just as a reminder to everybody, it's taking these patients over here and asking what happened over the course of a year of time. Did they progress and did they survive? And what we see here is really patients are being separated into a couple different buckets. For progression-free survival, you can see the blue and the red are also the patients that had the lowest number of genomes. So somewhere between 100 or less than 100 lymphoma genomes per milliliter. There's no statistical difference between these two lines, but there is a significant difference between that and patients that have more than 100. So more than 100 and up to 1,000. It's pretty obvious that if you are a patient with lymphoma, you want to have less than 100 lymphoma genomes per milliliter. It's not quite as clear-cut with overall survival, but it certainly is trending in the same way, where patients that have lower counts have better survival over time. And this is going out almost a couple years. But it's a very important takeaway here that the lower your residual disease, or the lower the count of disease before you start lymphodepletion, that is going to lead to better survival and better progression-free survival. That's probably intuitive, but here's the data to prove it. This is an appendix sort of data. It's not in the actual paper, but they asked the question, do ctDNA concentrations have any relationship to cytokine release syndrome and ICANS, which is another neurologic or neural-based, almost like a CRS. And don't want to spend too much time on it, but again, I think you're going to see, I think you can take away the same thing. These CRS grade 0 and 1, so patients that have low grade CRS, have at least on average, a 10-fold lower ctDNA concentration than patients that have grades 2 or 3 events. Again, just barely statistically significant. Similar thing with ICANS. Again, it's really just a spinal fluid based cytokine release syndrome. You're seeing a very similar pattern here where they have about 10-fold lower ctDNA than do patients with ICANS grades 2 and 3 and 4. And again, statistically significant. So not only do lower ctDNA concentrations lead to better progression-free survival and also overall survival, it also means you are less likely to experience a cytokine-related event. Okay, so this is a swimming lane plot of the clinical status and the MRD status in all progressors. Now, I want to highlight again, these are all the progressors. So these are the people that an event has happened. And I really like these kind of charts because it's kind of how the physician gets to see the patients. Let's just take a look at this bottom one because it's very easy to point to. A purple circle is a patient that's in a complete remission, and a brown square means they are MRD positive. You'll note that over time, so imaging, and then additional images and additional tests, imaging with circles or diamonds, and then the little boxes are MRD testing. Even though over time, this patient was MRD positive, you can see they didn't progress until day 360. So, this is actually a pretty good outcome if you're going to progress. That's giving a fair bit of time. There are a couple other ones worth noting here. Figure 1b, remember that progressor that we talked about? They started in a partial remission. We knew that they were MRD positive. They continued to test MRD positive, and by the time they reached day 90, they had progressed. So again, just another way of showing similar data, but we get to see it for all of the patients. I think the key takeaway with this slide is that all 30 progressors have at least one MRD positive finding at some point in their during their treatment. So really, the goal is to get to MRD negativity and stay there. Okay, moving on to figure 4, we're going to look at progression-free survival and overall survival based on their day 28 MRD and PET-CT imaging status. And again, I think what you can see is that the same kind of scenario is setting up here, where we have MRD negativity in the blue line or MRD positivity in the red line. So again, at day 28, and I kind of wish they'd moved the timeline in just a little bit here to make it a little clearer that this is day 28. So in this case, day 0 is actually day 28. It's a little confusing. But being MRD negative at day 28 is very statistically significant for any kind of progression event. And the same thing is true with survival. If you can't be MRD negative, the next best thing, right, is to have a complete remission by PET-CT, or if you don't have this testing available, ideally you want to have a complete remission by PET-CT, and you can kind of see the same thing. Somebody in a CR by PET-CT generally is less likely to experience a progression event than somebody who's in a PR, and the same thing is true with overall survival. Here we have some patients that talk about those. We've got day 28, somebody with stable disease, PET-CT, MRD negative. So they have disease, but it's stable. But what's interesting is that it's not able to be detected from a molecular perspective. And you can see at day 365, they have actually converted into a CR. Now, it's part of the wonders of biological science there. On the flip side, we have another person at day 28, stable disease, only this time they're MRD positive. In fact, if you compare this patient to the patient above them, they look to have very similar disease, same location, and about the same intensity of PET avidity. However, the clinical outcomes at day 90 are just remarkably different, because here they have progressive disease. You can see there's a lot of lymph node involvement in the neck and in the groin that was not there previously. So again, similar metabolic imaging results, but their MRD status really sets them down different pathways. Again, getting back to the idea that being MRD negative is really, really valuable. And it looks as though I have lost connectivity with my mouse. Okay, so this slide then looks at progression-free survival by MRD status, again from day 28, in patients with stable disease or partial remission. So in this case, all we're looking at is MRD negative versus MRD positive. And I think it's pretty obvious here that there's a very important and large statistical significantly different outcome in progression. Well, let's start that slide all over again, brother. Okay, here we go. Okay, so this slide is looking at progression-free survival by MRD status, again, by MRD status, from day 28, post-lymphodepletion, post-treatment, in patients that have stable or partial remission. So again, the blue line is MRD negative, the red line is MRD positive. I think you can see that there is a very, very clear difference in the rate of progression. It's much better to be MRD negative. In fact, using just MRD status and looking at PFS, we can use this laboratory test, or we can use the data to calculate some laboratory numbers. And so this test for these patients have a sensitivity and specificity of 94% and 82% respectively, and a positive and a negative predictive value of 88 and 90% respectively. So, you know, it's a pretty good assay, all things considered. Which brings us to endpoint review. Remember, there were two endpoints. The first, can ctDNA-based assessments at the time of starting lymphodepletion predict progression-free survival of patients with LBCL undergoing CAR-T therapy with at least a 6-month follow-up? The short answer is yes. And that was figure 2A, where we showed that patients were binned into those that have less than 100 lymphoma genomes per milliliter versus patients that have more than 100 lymphoma genomes per milliliter. If you have less than 100, it's quite predictive of what your PFS outcome is going to be. And then can the same kind of testing predict PFS at day 28? And that's the data that we just looked at. So the answer is yes. And it was very clear that just on MRD status alone, there is a 3- to 4-fold improvement in progression-free survival for those that are MRD negative. So congratulations to Adaptive, because the clinical trial met both of its endpoints, and I'm sure that this will be part of their application for IVD, for an IVD application. Okay, this brings us then to just some questions and some observations related to the study. I think the most important question, or the ones that your MSLs will get, is what does a physician do if MRD testing identifies a progressor? It's a good question. I think right now the answer is probably, you know, more poison. But it's possible that there are sufficient patients, or enough patients to design a progressor treatment study. And that would look kind of like what we have below. There's a common protocol where there's an MRD test. You bin these into MRD negative or MRD positive and MRD negative. This is kind of where our current study ends. But for those that are positive, they would go into another randomization event, and then some have intensified treatment and some have conventional treatment, and we'll see where the outcome goes. But obviously testing is going to be the requirement to be able to randomize these, to be able to include these patients in this study. I really did want to get back to my earlier comment about the higher than expected number of patients with mutations or double hits. And I personally think that this strengthens the results of this publication. By including more than you would expect, you would anticipate it being harder to achieve a difference in progression-free survival. And we know that because here's a Cohen 2014 paper where they're looking at single hits versus double hits, and you can see in both PFS and overall survival that there are, at least a twofold difference. So just by virtue of including more patients with double hits, I think it made achieving the endpoints harder. And again, I believe that this strengthens these results. The other kind of more sciencey question, but there was no conversation about whether the number of trackable sequences was associated with any kind of progression. I think it'd be great data if there was enough to talk about this. My inkling would be that yes, there is a relationship, but without any data, we don't know at this time. But it'd be really interesting if Adaptive was able to capture a little more data and comment on the number of trackable sequences and progression. With that, I'm going to say thank you very much for joining. I hope you enjoyed this presentation. Please feel to connect with us. Here's all of our contact information, and we wish you well. Take care.